Cellular
Respiration
by
Grant Campbell
and Jillian Cullen. To begin cellular respiration, you have to start by creating a very specific form of stored energy, ATP, which helps us grow, move, eat, etc. To use this energy, an ATP molecule, which is made up of 3 phosphate groups, ribose and adenine, must release one of its phosphate groups, which releases energy. Then an OH molecule takes its place. Through glycolysis, which occurs in the cytosol, 2 ATP are used to eventually create 2 more ATP and convert 2 NAD plus to 2 NADH. Glycolysis can act without oxygen through a process of fermentation, which allows glycolysis to start again, making it anaerobic. If the cell ends up going through an anaerobic process, then a carboxyl group from the pyruvate is released as CO2, while the rest latches onto a sulfur molecule, creating acetyl-CoA, which is temporary until the citric acid cycle. Through the pyruvate oxidation transition step, every pyruvate molecule from glycolysis goes into the mitochondrial matrix, the innermost compartment of the mitochondria. There, like we talked about before, it's converted into a 2-carbon molecule bound to coenzyme A, also known as acetyl-CoA. CO2 is released and NADH is generated. The acetyl-CoA made in the step before combines with 4 carbon molecules to create 6 carbon molecules, and goes through a cycle of many reactions. ATP, NADH, and FADH2 are produced, and CO2 is released. NAD reduces to NADH throughout, creating 6 NADH while releasing CO2. The NADH and FADH2 made in the last steps send their electrons in the electron transport chain, turning back to NAD and FAD. As electrons move down the chain, energy is released and used to pump out protons to the matrix. They go back through the ATP synthase enzyme, creating ATP. Oxygen then accepts electrons and takes up protons to create water.